de método das studies algo
All right. What happens to all the pyruvate that is made in glycolysis? They will go to the citric acid cycle for generating ATP if oxygen is available. If not, then we saw it is converted to lactate, and then the lactate is taken through the blood to the liver. Lactate is also produced by the red blood cells in large amount because that's their only means of getting energy by glycolysis. So that lactate also goes to the liver, and there it is converted back to pyruvate. So in the liver, then all the sources of lactate will be converted to pyruvate, and it is the same enzyme, lactate dehydrogenase, that initially converted the pyruvate to lactate in the muscles or in the red blood cells. So the enzyme is the same and the name is the same, irrespective of which direction the reaction goes. So in the liver it is all converted to pyruvate, and then what happens to that? So what happens to that is gluconeogenesis, which means making new glucose. So pyruvate will be converted back to glucose. So glycolysis was glucose to pyruvate, and gluconeogenesis is just the reverse of that, pyruvate to glucose. Now, as I mentioned before, all pathways go in one direction only. So if glycolysis takes it from glucose to pyruvate, the same pathway cannot take it from pyruvate to glucose. You have to have a different pathway. And as a general rule, whenever you have such opposite pathways, which is, this is not the only one, there are many other examples you will see. Whenever you have two opposite pathways, they will usually share the enzymes for the steps that are equilibrium steps or near equilibrium steps. And they will not share the enzymes for those that are the irreversible steps. So for example, in glycolysis, you know there are 3 irreversible steps, the 1, 3, and 10. So at each of those steps, so if you have pyruvate, it has to be taken to phosphoenol pyruvate. In glycolysis, it was the opposite direction. And in that direction, you make ATP because phosphoenol pyruvate is a high energy compound. So in glycolysis, you go from here to here and you make an ATP. And that happens whenever you are making an ATP or whenever it is an irreversible reaction. The same step cannot happen in the reverse direction. If we are going from here to here in one step, you cannot go from here to here in one step. So this reaction cannot happen. You can also understand that from an energy point of view. So if you plot the energy, let's say you are going from phosphoenol pyruvate, which is a higher energy, to pyruvate, and in the process you may be making an ATP or not making, but the question is, this is at a higher energy than the product. If you want to go in the reverse direction, if this difference is for making one ATP, providing one ATP is not going to take you there. Because remember, all reactions are irreversible here. So delta G is, first of all, it is highly negative and must be more than about 30, right? So because it takes 31 kilojoules to make one ATP. So the difference in energy between this one and this one must be more than 31, because some energy has to be released as heat energy. 31 will be used to make ATP, and the remaining will make the reaction irreversible. The definition of irreversible is that the delta G is highly negative. So that means the energy difference between pyruvate and phosphoenol pyruvate must be more than what is needed for making 1 ATP. So if we provide 1 ATP from here to here, that's not going to make the reaction happen, because that requires more than 1 ATP. And that is true for all irreversible steps. In any pathway, wherever you have an irreversible step, in order to go in the opposite direction using a different pathway, you cannot just provide 1 ATP if there was 1 ATP made in the other direction. So you have to provide 2 ATPs. And so in all these steps, then, instead of having this type of a reaction, pyruvate is first converted to an intermediate that is at a higher level than phosphoenol pyruvate, and then from there it goes here. So you need to raise it to a higher energy level so that it has enough energy to go down, and this step will also be a irreversible step. So that is true for all irreversible steps. If you are going in the opposite direction, you always have to provide more energy than what you got in the opposite step. If you did not get any ATP in the opposite pathway, then in order to go back, you need to provide at least 1 ATP to make that irreversible again. And usually, if this step is irreversible in one pathway, in the opposite pathway also this will be irreversible. And there is a reason behind it. The reason is that pathways are regulated at the same position. So if glycolysis is regulated at the pyruvate kinase level, then in the opposite direction also it will be regulated at that same step. And so regulation is always at irreversible steps. So this also needs to be an irreversible reaction. And in order to make it an irreversible reaction, you have to activate it first, and then it goes to phosphoenol pyruvate. So it will need 2 steps. So you cannot go from here to here in one step. So instead, you make another intermediate, which is oxaloacetate. And then you go to phosphoenol pyruvate. All right, so you need 2 steps, and that will require energy to do that. Oxaloacetate is made from pyruvate, so pyruvate structure is this, and this is converted to oxaloacetate using, oxaloacetate is a 4-carbon compound, so obviously you needed 1 carbon from somewhere, and that carbon is usually bicarbonate. And any time bicarbonate is used to add a carbon to any molecule, that enzyme will usually have a coenzyme called biotin. So the name of this will be, of course, pyruvate carboxylase, because you are adding a carbon dioxide. And the name of the enzyme is not so important, that it has a coenzyme, biotin. So that's true for all the reactions. You will see other reactions also where a carbon dioxide is added to a molecule to make 1 extra carbon. And the coenzyme for that enzyme will be biotin. Biotin is a vitamin, and you need it in very small amount. So no one is really deficient in vitamin, in biotin, unless you are on purpose not taking that biotin. One way of doing that on purpose is to take raw eggs in your food. Raw eggs has a protein called avidin that binds to biotin. So you should not take avidin in the natural form because it has a strong affinity for biotin, and that biotin doesn't get absorbed. This is the only way you can result in biotin deficiency if you consume raw eggs in your food. And why does the egg have this protein? Because this is another of their defense mechanism. They want to save the proteins and other nutrients in the egg from bacterial infection. And so they have this biotin so that bacteria cannot infect the eggs. But anyway, otherwise biotin is always available to everyone, so no one really has a deficiency of biotin. Now, whenever we are adding a bicarbonate, remember you are forming a bond with a carbon-carbon bond using carbon dioxide. That process is called carbon fixation. Whenever you take a molecule from the outside as a neutral molecule, as a molecule that is not bonded to any other carbon, and you are forming a bond to a carbon, that is a fixation, like nitrogen fixation. You form ammonia or any amine, or where you take carbon dioxide and form an organic compound, that is carbon fixation, and that is done by plants, right? So plants take carbon dioxide and make glucose. Humans are not known to make any compound from carbon dioxide, but we will see many reactions where carbon dioxide is actually used to make a compound. But always remember that we actually don't use carbon dioxide to make any compound permanently. So if that reaction is ever there, you can be sure that in the very next step or a few steps later, that carbon dioxide will be released. So although there are examples where carbon dioxide is used to form a bond, like it will form a bond here. The purpose of this is to preserve the energy in this bond. And in a later step, in fact, in the very next step in this case, that carbon dioxide will be released. So the carbon dioxide was added here just to preserve the energy from ATP. So of course it will need ATP in this bond. This is a beta keto acid. If you look at the formula of oxalic acid, this is a beta keto acid. So here is a keto group, here is a carboxyl group, this is alpha, this is the beta position. So beta keto acid, there is one thing that you should know about beta keto acids. You may have studied in organic chemistry that they can be easily decarboxylated. All beta keto acids are easily decarboxylated. In the cell, there are many reactions that do that. Like in citric acid cycle, there is a reaction that has a beta keto acid that removes the carbon dioxide. In the lab, in the test tube, even if you are not using any enzyme, you can decarboxylate a beta keto acid by simply heating it. So how does that happen? This COH is not important. The next. So this is the acid group, this is the keto at the beta position. And if you want to remove this carbon dioxide, this electron goes here and this bond goes here, and this oxygen gains the electron from this leaving carbon dioxide. So this is called an electron sink. In order to remove a carbon dioxide, you need an electron sink at the right position, and this is how it will decarboxylate without an enzyme, just by heating it. Of course, in the cell you don't heat it. In the cell it's catalyzed by the enzyme, but it is an easy reaction. So this reaction will remove the carbon dioxide, and that will give you a enolate ion. Remember, if you form a double bond here, that is you're forming an enol. Right, so if you are doing it in the lab, in a test tube without enzyme, this is the reaction. If the enzyme is doing it, this is still the reaction, except that you are using the energy of this bond, because remember, everything is about energy. So energy was used to form a bond to carbon dioxide. Now the energy of this bond will be used to form a bond to phosphate. So this will then react with the phosphate here, and that gives you phosphoenol pyruvate. Alright, so you make phosphoenol pyruvate in 2 steps of the acetate and then to phosphoenol pyruvate using energy from ATP, transiently forming a bond to carbon dioxide, and that carbon dioxide will be removed in the next step. In the later chapter, you will also find there are alpha carboxylic acids that are also decarboxylated. We have actually seen one, the pyruvate in the yeast. We were not doing that reaction in detail, but I mentioned yeast decarboxylates pyruvate to make acetaldehyde and carbon dioxide. That is also a alpha keto acid decarboxylation, which is a very difficult reaction. Beta keto acid is an easy reaction, enzymes are small. Alpha keto acid decarboxylases are more difficult reactions, enzymes are huge, they are multi-enzyme complexes with several coenzymes in them, so it is a more difficult thing to do because it's not forming an electron sink like this. So anyway, this is a beta keto acid that is decarboxylated to give you phosphoenol pyruvate. The idea is that, again, energy-wise, you need 2 steps. And after this, the rest of the steps up to the phosphofructokinase, if you remember, is equilibrium step. So this is step 10, then 9, 8, 7, I have the count right, and then you get fructose 1,6 bis phosphate. Right, so this is the next irreversible step. As I said, if you have 2 opposite pathways, the pathways will share the enzymes for the equilibrium step. So all these steps will be same as in glycolysis. Same enzymes, everything same about them. Just that they are in equilibrium, so they can go in either direction depending on which product or reactant is in higher concentration. And then when it comes to fructose 1,6 bisphosphate, that is converted to fructose 6-phosphate in glycolysis. This was the 3rd step in glycolysis, and the name of the enzyme, if you remember, you have a quiz, so you should remember what is the name of this enzyme. Phosphofructokinase, and that is PFK1. Because it has a number 1, that obviously means there is another enzyme called PFK2. And you will see that in a minute, what that is. So PFK1 converts this to this one, this is the energy investment phase. So if you want to go from here to here, it is again the same question. If 1 ATP was invested in this phase, you are not going to make 1 ATP going back in this direction, because you need more energy than that to make 1 ATP. The reaction has to be irreversible. The reaction is irreversible in this direction. The reaction needs to be irreversible in this direction also, and you need a different enzyme. You cannot share the same enzyme for the 2 pathways. And in this case, it is simply converted to here, and the phosphate is hydrolyzed by water. So as I mentioned before, high energy compounds by definition are those that will release a lot of energy when it is hydrolyzed, but in reality they are usually never hydrolyzed, because then the energy will be lost. And this is one example where it is actually hydrolyzed and the energy is lost. And there is a purpose behind losing that energy, and that is to make this reaction also irreversible. As I said, for 2 opposite pathways, if one has an irreversible step, in the opposite pathway also there will be an irreversible step at that same position using a different enzyme. So what will be the name of this? It's you are removing a phosphate, so it will be a phosphatase. So it will be fructose 1,6 bisphosphatase. And then from here, again, there is another equilibrium step that gives you glucose 6-phosphate. And then again, step 1 was an irreversible step. So in glycolysis, glucose gives you glucose 6-phosphate, adding 1 ATP. There was also 1 ATP here. Which enzyme? So this is going back, fructose 1,6-bis phosphatase. Because you are removing a phosphate. If you are adding a phosphate, then those enzymes are called kinase. Removing a phosphate, those enzymes are called phosphate. So in glycolysis, the first step was conversion of glucose to glucose 6-phosphate using 1 ATP. And once again, if you are going back in the opposite direction, you cannot be making 1 ATP because then it will be barely matching the energy. The energy has to be actually higher to make it more irreversible. And so again, in this case, the energy, this is simply hydrolyzed. So again, water reacts with it. So these are two examples then where a high energy compound is hydrolyzed, that the reaction is irreversible. And once again, what will be the name of this? This is a phosphatase, so it will be glucose 6-phosphatase. When we did glycolysis, I remember I mentioned one purpose served by this reaction is to prevent the glucose from leaving. If glucose enters the cell, you don't want it to leave the cell. And so you put a phosphate on it, and that phosphate will later be used to make ATP, but another purpose it serves, that the phosphate cannot leave the membrane and go out. That way the cell keeps the glucose inside. So in this case, that phosphate is hydrolyzed, and the very purpose of hydrolysis of that is to export the glucose outside. So this type of reaction, gluconeogenesis, doesn't happen in all cells. It mainly happens in the liver. As I mentioned before, liver works for the rest of the body, and the purpose of gluconeogenesis is not to use the glucose for its own needs. It also has its needs, but it makes more glucose than that, and that is exported to the rest of the body. So in order to do that, you have to remove this phosphate, and then the glucose can go out of the membrane into the blood and be transported wherever needed. All right, so as I said, for every irreversible step, there will be another irreversible step in the opposite pathway also, and one purpose of that is the regulation. And you want to regulate both of these enzymes at the same time. You want to turn one of them on and another one off. That type of regulation is called reciprocal regulation. So reciprocal regulation means the same regulator turns one pathway on and another pathway off. And obviously the purpose then is you don't want both of them on at the same time. What will happen if you have, and let's say, an inefficient regulation and both of them are on at the same time? What will be the net result? It will be a cyclic pathway, right? You are going from here to here and then from here to here. So this is a small cyclic pathway. And when that happens, that is called a substrate cycle. Substrate cycle is also called a futile cycle. Because you don't achieve anything, right? You start with one, go to the other one, and then another one goes back to this one, so you go back in circles. What do you achieve? In every cyclic pathway, that is true. You start with a compound and you end with that same one. But you achieve something. Like in citric acid cycle, as we will see, its purpose is to convert two carbons of acetate carbon dioxide. We will study that later. So every cyclic pathway has a purpose, even though you are going in circles, there is a purpose. So what is the purpose of this? What is the net result of this? Net result is that you are hydrolyzing an ATP. You're hydrolyzing an ATP here, then you're adding another ATP and again hydrolyzing that ATP. So you're basically, the purpose of this cycle will be to hydrolyze ATP. And that will be a waste of energy. You're not achieving anything, and that's why it's called a futile cycle. So does it actually happen? If your regulation is good, then it won't happen, or should not happen. Whether it actually happens in humans, that is debatable. Some people say it does, and that's why people lose weight. Some people lose weight more easily than others, but there is no evidence that the substrate cycle actually is causing weight loss. So in humans, most likely this is tightly regulated. So you don't have both of them on at the same time. But then there are other animals that have this substrate cycle on purpose. So it has evolved to have the substrate cycle on, and what is the purpose of that? To generate heat without doing any work. That means when animals hibernate in winter, they have their substrate cycle on. Not just this one, there are other substrate cycles also. They have them on so that heat is generated by hydrolysis of it. Alright, so this regulation at this point is called reciprocal regulation by the same regulator, and the same regulator in this case is by fructose 2,6 bisphosphate. There's another molecule called, this is the fructose 1,6 bisphosphate. The cell also makes a small amount of fructose 2,6 bisphosphate. So that means some of this fructose 6-phosphate is shuttled here to make this one. You need very small amount of it because it's a regulator, so you don't need a lot. So small amount of fructose 6-phosphate will be converted to fructose 2,6 bisphosphate instead of 1,6. And of course, the name of that enzyme will be phosphofructokinase, and that is the 2. So this is PFK. So as I said, if this is PFK1, there must be a PFK2 somewhere, and this is the enzyme that makes the regulator for that pathway. And it of course makes it in a small amount because the regulator, you cannot be using a lot of it. So it is just a binding. And this one will turn, if this pathway is turned on, then this will be turned off. If this is turned off, this is turned on. So it will turn on one pathway and turn off another pathway. So that is true for all substrate cycles. There will be a regulator. We will see other examples also where the same enzyme is turning on one pathway and turning off another. Alright, so that is the gluconeogenesis, or making glucose. And as pharmacists, you should know that this is the pathway that is targeted by drugs for diabetes, like metformin, that inhibits gluconeogenesis, and that's how you prevent synthesis of glucose for the patients. So what are the precursors of gluconeogenesis? So one of them, both two of them, as I mentioned here, one is the lactate, pyruvate. And anything else that will feed into the glycolysis pathway or the gluconeogenesis pathway. There are so many steps. If you can make something that will enter here, let's say, then this will also be a precursor for gluconeogenesis. So anything that makes an end product that is in common with any of the intermediates of glycolysis pathway, that will also serve as a precursor. One example of that, so besides lactate and pyruvate, these are the main ones. Another minor one is glycerol. Everyone knows where glycerol comes from. We will study lipids later. Lipids have fatty acid linked to glycerol. And also we will study that lipids cannot be converted to glucose. So as pharmacists, you know that people always want to gain weight, lose weight or whatever. You can convert proteins to carbohydrate. All the excess protein you eat will be converted to carbohydrate and to fat. Excess carbohydrate you eat will be converted to fat. So they go both ways. But fat cannot be converted to carbohydrate. Proteins can be converted to carbohydrates. Carbohydrates can be converted to proteins, except for the nitrogen. The nitrogen must come from somewhere else. And carbohydrates can convert to lipids, but lipids cannot be converted to carbohydrate. And we will see why that is so later in another chapter. But the slight exception to that rule is that a small portion of the lipid is converted to glucose, and that small portion is glycerol. So lipids contain glycerol plus fatty acids. If you compare the number of carbons in both of them, the glycerol probably constitutes about 5% of all the carbons in lipids. So 95% of lipids cannot be converted to carbohydrate, only 5%, which is the glycerol portion, that can be. So glycerol is a 3-carbon compound. Alright, and by the way, as pharmacists, you should also know that glycerol is the same as glycerin. The same thing, glycerin is a component. If you read the label in many lotions and they will say glycerin, it's the same thing as glycerol. So glycerol, of course, you put a phosphate here. To make glycerol 3-phosphate, so that the enzyme will be glycerol kinase. With ATP, you add a phosphate to the 2 position, glycerol 3-phosphate, and then you oxidize this one, this OH into a keto group. And what is the name of this product? This is the intermediate that enters the glycolysis pathway or gluconeogenesis pathway. What is the name of this?
G3G.
This is dihydroxyacetone phosphate, DHCAP. So to be a precursor of gluconeogenesis, all you need to do is to take it into one of the intermediates of glycolysis, and then the rest of the steps from there will be the same as the gluconeogenesis. So the cyclic pathways, the ones that I mentioned, are happening all in the same place. But cyclic pathway can also involve 2 different parts of the body. So half of it happens in one organ, the other half happens in another organ, and that's what we actually saw here. So for example, in glycolysis, pyruvate is converted to lactate in muscles. When you are doing exercise, then the lactate goes to the liver. There the lactate is converted to pyruvate and then to glucose. And where did this pyruvate come from? It came from glucose. This will happen in the liver, but the liver doesn't really need all that glucose, so what will it do to the glucose? It will send it to the blood by this reaction, make it into glucose 6-phosphate, converts to glucose, and then that goes back to the muscles. So it doesn't go back only to the muscles, it goes everywhere, but some of the glucose will go to the muscles, and so that involves a cyclic pathway. Glucose to lactate and then lactate to glucose, you are starting with the same molecule, ending with the same molecule. So whenever you have a cycle like this, that is not easily discovered because they are happening in two different places. But this was discovered by Cody. So this is called the Cody cycle. So once again, the same question arises: what do you gain by this? Any cyclic pathway, you start with the same thing, end with the same thing. So what do you gain or lose by that? What is achieved in this cyclic pathway? So what is achieved is this: in glycolysis, you make 2 ATP. In the whole of glycolysis, you make 2 ATP. If you want to convert like glucose, pyruvate to glucose, how many ATPs do you need? You need, of course, you need more than 2. How many more? It will be 4 more. 1 ATP is lost here by hydrolysis, 1 lost here by hydrolysis, and in order to activate it, you need 2 ATPs. Remember, in order to go from here to here, you have to add more than 1 ATP higher to get it to a higher level. So that means you need 2 ATPs there. So there are 4 ATPs needed to do this. So 6 ATP. So you need 4 ATP extra. That means this cycle is burning 4 ATPs. Whenever you are doing exercise on a machine, it tells you how many steps you ran, how many calories you burned, how many fat calories you burned. But remember, the fat calories that it says there is not the one that you actually burned. That's the amount that you are going to burn. Well, when the pyruvate goes to the liver, is converted to glucose, you use this 6 ATP that comes from oxidation of fat. Alright, so the number, don't be excited by the number that you have already burned that calorie. You are going to burn that calorie when you are actually not doing exercise and your body is still working. Convert all the pyruvate to glucose, and that's where you burn the fat calories here. The extra 4 ATP for glucose. There is another cyclic pathway that is involving glucose and pyruvate, and that is called the glucose-alanine cycle. We will study this cycle actually twice. One will be now and another will be when we study amino acids. Because the purpose of this cycle is twofold. One, it is a cycle, so we start with glucose and we will actually end with glucose. The other purpose is to remove the nitrogen of amines and take it to the liver. So if you have excess proteins, as I said, proteins will be converted to carbohydrate. But carbohydrates don't have nitrogen. So what will happen to all the nitrogens from all the amino acids? That needs to be deaminated, and that ammonia has to be transported to the liver, not as ammonia, because ammonia is very toxic. So it has to be transported in another form that is not toxic, and then it goes to the liver. In the liver, the ammonia is converted to urea, and that is excreted in the urine. So purpose of this cycle is twofold. One, again, you are starting with glucose, ending with glucose, and you are transporting the nitrogen of all amino acids that need to be degraded. You are transporting it to the liver for converting into urea. So if you have any excess amino acid, whatever amino acid, they will be converted to the corresponding keto acid. Remember when we did the enzymes, we talked about transaminase reactions. Every amino acid has a corresponding keto acid. If you transfer the amino group to something else, it becomes a keto acid. And it is transferred to another keto acid that becomes a corresponding amino acid. So in this case, the pyruvate is the keto acid of alanine. Once again, pyruvate is the corresponding keto acid of alanine. So alanine, you just put, instead of keto group, you put the amine here. So if you put amino group from any amino acid into pyruvate, it becomes alanine, and that's what happens in the muscles and in all cells where you have excess of amino acid. So that will be all converted to the keto acid, and pyruvate will be converted to alanine. What will happen to this keto acid? That's the whole idea of converting your excess protein into carbohydrates, so that will go through other steps to get into carbohydrates and fat. And this alanine, which is carrying the amino group from all these excess amino acids, will go to the blood and then to the liver. So alanine is transported to the liver, where it will be converted back to pyruvate, and the nitrogen will be, I'm writing it as ammonia, but not really so. There is another receiver of ammonia. Ammonia is formed, but there is another amino acid that takes that ammonia. That we will study in the urea cycle in amino acids. So the idea here is that all the amino groups of all the amino acids are eventually transported to the liver for converting into urea, and then you end up with pyruvate. So you started with pyruvate, which came from glucose, and the pyruvate will also be converted to glucose. And then the glucose, the liver doesn't keep much of the glucose, so it will be distributed in the rest of the body. So that's another cycle involving more than one organ, and that is called the glucose-alanine cycle. And as I said, this achieves two things. One, again, you will be consuming 4 ATPs extra in order to go make the glucose from pyruvate, and you are going to transport all the amino groups as to ammonia in the liver to be converted to urea. Alright, so that is gluconeogenesis. The next thing we are talking about is glycogen. Glycogen synthesis and degradation. Glycogen is stored in the liver and the muscles. One more thing about the gluconeogenesis, as I said, the last step, the hydrolysis of the glucose 6-phosphate to glucose takes place in only the organs that carry out gluconeogenesis. One example was the liver. There are others also that do that. For example, the kidneys do it, small intestine does it. So it doesn't happen in all the cells. There are only some cell types that carry out gluconeogenesis. So in the same way, glycogen is stored in the liver and the muscles for future use. As I mentioned in the first class, the amount of glycogen that you have in your body is only good enough to last for 24 hours. So if you are fasting for 24 hours, you have probably used up all your glycogen. And then you need another source of energy. So to break down of glycogen, we already saw this is a structure similar to starch. So you break down from the non-reducing end. So if this is your glycogen with branches, so that will break down glucose from the non-reducing end. This is the reducing end, and these are all non-reducing ends. And as I mentioned, if you do the calculation, you will find that you can get quick energy from about 50% of all the glucose units. Because glycogen is 10% branched, that means every 10th unit, glucose unit, there will be a branch. So glycogen phosphorylase is the enzyme that breaks down glucose from the non-reducing end up to the fourth glucose, 1, 2, 3, 4. So up to here it will remove one glucose at a time and stop at the fourth glucose from the branch. And that it does from all these branches. So if you do the calculation, you will find that you can get quick energy from glycogen in the muscles by removing about 50% of the glucose units by glycogen phosphorylase, which is a fast enzyme, so it gives you glucose quickly, and that's why athletes who run, they run at very high speed for maybe 10 seconds, when all these 50% glucose are all broken down during that time. And then the remaining steps will be slower because so I'm drawing only one branch maybe, so you have 4 here and 4 here. So it will stop at this, and if you do that for all these other branches, that is called limit dextrin. So getting from glycogen to limit dextrin is a fast process using glycogen phosphorylase. And then there will be a debranching enzyme. Debranching enzyme, the same enzyme has 2 activities. Alright, so one of them is to remove three units from here and transfer to the nearest branch. So these 3 units will be removed from here. That means you will have only one glucose left, and you already had 4, and then you have another 3, so that will be 7. Alright, and then again the glycogen phosphorylase will remove all these up to the 4th one, and this way the process continues. What happens to this one glucose at the branch point? Remember, at the branch point, this glucose has 2 glycosidic bonds. One will be a alpha 1,4 and another is alpha 1,6 because of the branch. Because it has 2 glycosidic bonds, these enzymes don't recognize it. Glycogen phosphorylase cannot break that bond. And so this is removed by the second activity of this one, which is a glucose 1,6 hydrolase. So it basically removes this one piece of glucose as a glucose monosaccharide without adding a phosphate on it. Remember, in glycogen phosphorylase, whenever you are removing all these 4 glucose, it comes out as glucose 1-phosphate. So glycogen is glucose alpha 1,4 glucose. The linkage is with the carbon 1, and so the phosphate is added to carbon 1. So the product of all these is glucose 1-phosphate. The product of this one is glucose. So that's the one that is hydrolyzed. This is another example where you have a high energy compound, not just one bond, but two glycosidic bonds. But we don't have the right enzyme to take care of that, and so that is simply hydrolyzed, and so you don't get that extra energy. Yes. Number 2 is a hydrolase. Hydrolase. It means this bond is broken and damaged. And glucose comes out as just free glucose instead of glucose phosphate. Yes. 1,6. That's a branch, right? Hydrolase. Hydrolase means it is hydrolyzed by water. So once again, high energy bonds are not really usually hydrolyzed, but some cases they are because there is no other option. So here there is no other option. There is no enzyme to take care of it, and so it is hydrolyzed. That means you are not going to get the extra ATP from here. How many ATP will you get from glucose that comes from glycogen? Glycolysis gives you 2 ATPs, right, for glucose. Glucose that comes from glycogen, you will get 3 ATPs and not 2 because it comes with a phosphate. There is already energy in all these bonds. When the glycogen was made, energy is already incorporated into all those glycosidic bonds. So when you are hydrolyzing those bonds and forming glucose 1-phosphate, you are already having a 1-phosphate in that. So instead of doing the first step of glycolysis, you skip that. If the energy is coming, if the glucose is coming from glycogen. However, that applies only to the muscles. Remember, glycogen is in the muscles and the liver. The muscles use it for themselves. Whenever you are doing any work, mechanical work, muscles will use that glycogen and they will get 3 ATPs per glucose. On the other hand, the liver is storing the glycogen for the rest of the body. So when the liver breaks down glycogen, it will hydrolyze that glucose 6-phosphate to make glucose, transport it into the blood, and then it goes to the rest of the body. So glycogen from liver gives you again 2 ATPs per glucose. Glycogen from muscles gives you 3 ATPs per glucose. But it is not exactly 3, it is slightly less than 3. Why is it slightly less than 3? Yes?
Because you still make that one glucose?
That one glucose, if you calculate, it's 10% branches, and one glucose is hydrolyzed without getting that extra ATP. So instead of 3 ATPs, you get 2.9 ATP per glucose. Alright, so glycogen from muscles gives you 2.9 ATP. That 0.1 difference is because of that one glucose and in every 10 branch point that is simply hydrolyzed. And glycogen from liver will again give you only 2 ATPs per glucose because they will be transported to the other cells in the body. Alright, so that was the glycogen degradation. We also need to talk about glycogen synthesis. We will stop this at 10:30 when we will have the quiz. So now probably is the right time to get your 5-minute break to start with glycogen. That is the same thing that you have
You remind me about every.
Oh my god.
And you get upset every time. So I stopped. No, well, I forgot
was
it in my
sleeping all
pocket.
day and I felt like shit. And I felt like dead by the time I got home.
Why are you guys so far?
And the one time they still do it.
Who's on the other side of you? Is there an empty set?
They're like, oh, I'm stuck here.
But is there an empty? I'll just walk out.
Because when I active practice, I don't want to talk about scoring that. You get upset. What's the difference?
What do you mean, what's the difference? I'm not in school. I'm in school mode. I'm in practice mode.
Wherever it might be that.
I'm gonna get like
It's like registering.
Because it doesn't. It's all I talked about for 2 days, and I didn't mention it on Sunday.
when did I Right, well that was gonna talk about something that I'm just happy what you wrote later and
سألت سؤال وطلعت ماني فاهم. The steps are, he didn't number them here, but I was going based on so I was just, but there was one step that he wanted that I couldn't find was missing. It's like, what step number is this? I went one by one. I don't know anything about it.
But like, what if the question's like which? Oh yeah, which
I just asked him about the exam. He said, I don't know. I said, how many questions? I don't know. I didn't make it. Okay. Okay.
It's okay. But like, I might be more stressed for that than anything. Like I can't see it one. That's crazy. No, that's
I did that too.
I
Whoa, the switch on.
That's okay, so that's good. That's good to hear.
That question is
It's not wrong. When was I ever on your side? Was I ever on anyone's side?
He's like, I think
I'm not even on your
And lying through your teeth, but you're smiling like a jerk. Ryan, I've known you since middle school. I know when you're lying and you're telling the truth.
Except for you. Except for you. He was like, hey, where's that tall Egyptian boy? And he was talking about you. No, we
Roman, do me a favor. Can you smack him on top of the head for me? Do it, please. Thank you.
I'm afraid for my safety now. You heard about that?
Sorry. I was watching them. I was crazy. It was by the third one. It was when that guy struck Two strikes. The guy was it had been down the middle. I don't think he could. You know, it's
Alright, so is everyone back? Alright, so for glycogen the opposite of glycogen degradation. So this is another example of the opposite pathway, which again means that the equilibrium steps will be using the same enzyme, whereas the irreversible step will use a different enzyme. But here we will study too many enzymes. There are degradation doesn't use so many enzymes. So what will be the first step of glycogen synthesis? You are starting with glucose. So you are starting with glucose that will be converted to glucose 6-phosphate. Same step as glycolysis. And then the glucose 6-phosphate needs to be converted to glucose 1-phosphate. Remember, that's what came out from the glycogen, because the glycogen means it is the alpha 1,4 linkage, so the phosphate goes to carbon 1. So if you want to make that compound again, then you have to convert 6-phosphate to 1-phosphate, and what will be the name of this enzyme? Whenever you are transferring a phosphate from one position to another, that is called a mutase. The mutase, the phosphoglucomutase. And then this will be used to make glycogen. And of course, you cannot use the same enzyme because those steps were irreversible steps. So you need different enzymes to make it. And energy-wise, you have to raise it to a higher energy level so that it can come down to a lower energy level and make the step irreversible. And that process is called activation of the substrate. So first you need to activate the substrate, which in this case is glucose 1-phosphate. And you need to convert it to an even higher energy compound. And for that you need ATP, which in this case is not ATP but UTP. So as I said before, all these nucleotide triphosphate, they behave the same way, they have the same amount of energy. So in many reactions you will find instead of ATP, it is UTP. UTP is the next common one commonly used for energy. So UTP will be protein, ribose, and then 3-phosphate. This is UTP. What's the U? U is uridine, which are the 4 nucleotides that make up DNA and RNA. One of them is uridine. And so just like adenine, there's uridine. Uridine has 3-phosphate, just like ATP has 3-phosphate, and it works exactly the same way. It is a high energy compound for same reason that we discussed for ATP. So why does it use UTP? That's how the enzyme has evolved. So many of the enzymes use this one. This is the second most commonly used high energy compound for energy. So what that gives you is UDP glucose, uridine, ribose, then phosphate, phosphate. As I mentioned, you have to raise your energy to a high level and then you come down. So this is the high energy level with this one, uridine. So this is called UDP glucose. Uridine diphosphate and then that's glucose. What will be released? What is the other product of this? There are 4 phosphates here, right? 3 plus 1, 4. You have 2 of them here. That means the phosphate other product will be pyrophosphate, PPI. Pyrophosphate. If you want to know the structure, it is just phosphate into another phosphate. So that is pyrophosphate. And this is very important. Pyrophosphate you will see many times in many reactions. This is a common strategy used by many enzymes to make pyrophosphate. Whenever that happens, you can be sure that this reaction that is written here has a delta G goes to 0. That means this is not a highly negative delta G, or this reaction is not really feasible, it barely happens. But you cannot really have enzymes or steps that can barely happen. You want all the steps to be irreversible steps. So this is the irreversible step in the glycogen degradation. So in the reverse direction also you want an irreversible step. And that's where the pyrophosphate comes in here. Pyrophosphate is never present in any cell at high concentration, because the cell always has an enzyme, pyrophosphatase, which is always present, and anytime pyrophosphate is formed, it will hydrolyze. And this is the strategy used for not just this enzyme, many enzymes you will see later also, pyrophosphate is released, and you can be sure that at the next moment that pyrophosphate will be hydrolyzed. And what is the purpose of hydrolysis of the pyrophosphate? This is the Le-Chatelier's principle that says if you remove one of the product, then the reaction moves forward. Or this is also called, you are coupling to reaction. This is a coupled reaction. This reaction has a delta G0, hydrolysis of pyrophosphate has a delta G, I don't remember how much, but something like minus 40, let's say. Much higher than, it's a little higher than the phosphate. So that means this energy then will be combined. So this is minus 40. So the net result is these two will have a delta G that is highly negative. So the purpose of having pyrophosphatase is to drive a reaction forward, which otherwise wouldn't go forward because they have a very negative or 0 delta G value. You can look at it in another way. When pyrophosphate is converted to phosphate, and what will that phosphate be used for? Later on, it will be used to make ATP, and that means you will again have to form another phosphate bond, another bond to ATP. So basically, you are using two ATPs here, or two equivalents of high energy compounds. Whatever the name is, one name is UTP, the other name is pyrophosphate, but they are similar to ATP. You are using two equivalents of ATP with different names. Alright, and that's the whole idea again here, that you want to raise it to a higher level. In order to do that, you need more ATP than what is generated in the degradation step. So that's why you need two equivalents of ATP. So I'm writing two ATP, but none of these is ATP. One is UTP, the other is pyrophosphate, but it's two equivalents of ATP. Same with the previous one that we saw, going from pyruvate to phosphoenol pyruvate, you needed two equivalents of energy to form that bond to carbon dioxide. We always need two. If in the reverse direction, it gives you one ATP. So with this UDP glucose, then you can transfer the glucose, because it is at a higher energy level, you can transfer it to glycogen with another glucose. That means you already have a glycogen with a n number of glucose in it, and you are adding another glucose molecule to it, so you are with n plus 1. So when glycogen is synthesized, you are always adding the glucose to an already existing, like you have a question? No. To an already existing glycogen. So you need a glycogen to add another glucose to it. You cannot, the enzyme cannot really add one glucose to another glucose to form a disaccharide. So it doesn't make a disaccharide. It will add a glucose to an already existing polymer. So that is called a primer. So if you have this one, UDP glucose plus already existing polymer, which is also called a primer, and then that will give you UDP plus glucose with n plus 1. That means you have added one glucose in it. Alright, so this way then, so this enzyme is called glycogen synthase. So in other words, the important enzyme in the opposite direction of degradation is glycogen synthase, and in degradation it was glycogen phosphorylase. Just like as I said, whenever you have two opposite pathways, the two enzymes in the two pathways will be regulated simultaneously by the same regulator. So degradation and synthesis is regulated by the same regulator, and so this is another example of reciprocal regulation. And once again, you don't want both of them to be on at the same time. That doesn't achieve anything. So the regulation must be very tight. When one pathway is on, the other pathway must be turned off. So this primer, as I said, there must be already existing primer, but then I also mentioned when glycogen is degraded, after you get to the limit extreme, then with time you can degrade the whole glycogen. Within 24 hours, all your glycogen is gone. So if all your glycogen is gone, how do you get a primer? Where do you get a primer from? Where do you start the first glycogen synthesis? So when I drew the glycogen structure, there was one reducing end, and then there were many non-reducing ends with these branches, but there was one reducing end that I keep mentioning all the time. But that is also wrong, because that reducing end is not there. Why is it not there? Because the first glucose that is added to a primer, and the primer is an enzyme or a protein called glycogenin. So glycogenin is the primer for adding the first glucose in glycogen synthesis. The first glucose is added to a protein called glycogenin. So that means every glycogen molecule, whether it's a liver or muscle, has a glycogen at the reducing end. So there is really no reducing end of glycogen. There are many non-reducing ends, and at the reducing end that is also blocked by a protein called glycogen. This is also an enzyme that actually catalyzes its own reaction with the glucose. The first glucose and then a couple of glucose are added to it also by the same enzymes. About 8 or so glucose are added by glycogen into itself. And that creates a short glycogen polymer of about 8, which now acts as a primer for new glycogen synthesis. So then the glycogen synthase will keep adding the glucose to that small primer. So reciprocal regulation of glycogen synthesis and degradation will be at this step, where you have glycogen synthase and glycogen phosphorylase. So I sent you some figures, and if you later on if you see this figure there, that shows the regulation of glycogen synthesis and glycogen degradation. So glycogen phosphorylase will exist in two forms, active and inactive. So this is how the regulation happens. This exists in two forms. One will be the active, let's say we call the active as A and the inactive as B. So when the active form is converted to when the inactive form is converted to the active form, it will degrade glycogen. So that means there has to be I'm drawing two arrows, means they are converted to one. It's not an equilibrium, either it goes this way or the other way. In the active form, the glycogen phosphorylase is itself phosphorylated. As I mentioned before, one way of regulation was allosteric regulation, another method is covalent modification, and usually the covalent modification is adding or removing a phosphate. So this is one example of covalent modification or regulation. If this enzyme adds a phosphate, it becomes active. If this enzyme is dephosphorylated, then it becomes inactive. So obviously the enzyme in this direction will be a phosphatase, in this direction will be a phosphatase. So the enzyme is converted from active, inactive or inactive to active by phosphorylation, dephosphorylation cycles. So there will be an enzyme called, I am not naming the enzyme, let's say it is a kinase. And the kinase will activate this, that means it will put a phosphate here. And there will be a phosphatase that will activate this one, that means remove a phosphate and make it inactive. Alright, so this kinase then activates one, inactivates the other pathway. As I said, you cannot have both pathways on at the same time. So the kinase is an enzyme that cannot be a regulator. Regulators are usually small organic molecules. And the regulator in this case is the hormone insulin. So the insulin will activate the kinase, and then kinase activates in this direction. Phosphatase is activated by a different hormone. So if phosphatase is Alright, so the insulin activates the phosphatase and the phosphatase removes the phosphate and gives glycogen phosphorylase. If this is active, then this will degrade glycogen. On the other hand, the opposite pathway will be glycogen synthesis, and for that you need the enzyme glycogen synthase. So there is another enzyme glycogen synthase. Needs the differentiation. Which also exists in two forms, with a phosphate and without a phosphate. So in this case, the glycogen phosphorylase was active when it is with the phosphate. The opposite is true for glycogen synthase. It is inactive when the phosphate is there. So that means this is the active form, and that will make synthesis. So when this is active, it will synthesize glycogen. When this is active, it will degrade glycogen. And you cannot have both of them active at the same time. And that's why phosphatase will remove this phosphate. Sorry, this is the opposite arrow. This arrow goes this direction, this arrow goes this direction. So phosphatase removes the phosphate and makes it active. Kinase will add the phosphate and make it active in the opposite direction. So glycogen degradation is activated by the kinase and inhibited by the phosphatase. So phosphatase enzyme inhibits glycogen degradation. Phosphatase enzyme activates glycogen synthesis. And in the same way, the kinase enzyme will add a phosphate to this and activate degradation, and the same enzyme, let me draw it this way, will convert glycogen synthesis to the inactive form. Alright, so once again, glycogen cannot be made and synthesized at the same time. So to regulate that, insulin will activate the phosphatase, which converts glycogen synthesis to the active form. And so glycogen will be made. What is the purpose of insulin? To lower the glucose level. How does it lower the glucose level? By making glycogen, converting glucose to glycogen. So insulin activates phosphatase. Phosphatase will remove a phosphate from the glycogen synthase and start making glycogen. The same enzyme will also remove a phosphate from the glycogen phosphorylase and make it inactive. Alright, the phosphatase removes the phosphate from this and makes it inactive. The same enzyme makes the synthesis active. And then there is this kinase, which does just the opposite. And what will regulate the kinase? Just like insulin was regulating the phosphatase, so there is another hormone, glucagon, that will activate the kinase, and kinase at the same time will activate this enzyme and inactivate glycogen. So this is an example of two reciprocal regulations happening, one with insulin and another with glucagon. So glucagon increases the blood glucose level by activating glycogen degradation. Insulin decreases blood glucose level by activating glycogen synthesis, and that is done by activating and inactivating the two pathways simultaneously. So that is the example of reciprocal regulation again. And that completes this pathway. So the only other pathway related to before the midterm, which will do on Thursday, is the chemical switch pathway. And then the rest of the time we will be doing things that will be so for the there was a lot of promotion at the end for people that are walking all the way here. I hope you can stay here for 10 minutes and then end at the same time so that everyone